Good morning, everyone. So my name is Michael. Um, for my presentation will be a deviation, so to say, from the previous speaker. So I'm from the pure science side. And basically, my focus is on study of fishes, inventory of fishes in different rivers, and giving them scientific name and other implications of that. So today I'm presenting on a project we did and still ongoing. So I will start from explaining so that we can have good background to what I'm presenting. So rivers are classified based on the fact that fishes found in them can be unique and exclusive. So in Africa, you have these classifications. And then specifically in Nigeria, you have the geographical region of Nigeria overlap with two of these ichthyological classification. So in Nigeria, that means you have all the rivers grouped into two in terms of the fact that the fishes found in them are unique to each of them. So when you look at Nigeria geography now, so you can see there's the Nilo-Sudan province and there's the Lower Guinea province. So it means the fishes found in the Nilo-Sudan province are basically different from the one you find in the Lower Guinea province. So the problem, scientific problem, is that the Nilo-Sudan has been like almost completely studied, especially compared to the Lower Guinea province. And so the focus of the project then, understandably, is the Lower Guinea province. So then again, when you look at the Lower Guinea province, I don't know whether I can can this point? I don't know. Okay, if you cannot point, the other map, there's the red portion there, and that red portion, so is the easternmost part of the Lower Guinea province relative to the Cross River. So that part, it's even like almost completely unstudied. If you look at it, you see other points that have been studied at the Lower Guinea province. But there's more or less no record of fishes from that eastern part. So, and that's the focus for this study. So, like I've given, so we inventory the fishes of the area. So basically in this study, the focus was more of the shrimps because the diversity, or if you look from the economic point of view, people will not focus on studying small species of fishes, but large species of fishes. So like the main fishing gear I used was what I was holding. And then when fishes are sampled, when they are collected, we take pictures of them underwater to keep the live coloration, because after collection, the next thing is to preserve them, meaning formalin, and after you preserve them, the coloration is lost. So you want to retain the life coloration, and then we take other data like the location data, the vegetation data, and the rest. And ultimately, we also take samples from the fishes in order to do DNA analysis. So in this case, there was COI DNA analysis and also whole genome DNA analysis. Basically, in order to, so you've seen the fish, you want to be very sure of their scientific identity, and that's why you run the two kind of analysis. In total, for the period, so I ran it during the dry season in Nigeria, so 2020, 2022. So I had short period. You see, it's a large geographical region, short period in order to, short period, so that's why it's run between 2020, 2022, and 2024. So in total, there were 700 fishes collected. So I'm not going to go into deep scientific. Yeah, so at the end of the day, there were 33 fish species documented. But the major discoveries, so number one, so I will use the word tilapia. So there were 4 tilapia fish species that were not known to science that was discovered. And then number 2, there is also another fish species that is called Procatopus. So at least 2 of them, because the project is still ongoing, is also not known to science. Then there is also another species called Epiplatis. So 1 of them not known to science. The last thing talks about confirmation of presence of Fundulupacha indianus and Fundulupacha skili. So in that geographical region, there's what we call terra typica. That is, the river, each fish species was first collected. That's terra typica. The river where each fish species was first collected. So in that geographical region where I survey, so there were terra typica. So we were able to locate the terra typica of Fundulupacha indianus and also Fundulupacha skili, and we were able to collect them again. So the significance of it is that when they were originally collected, they didn't take sample for DNA documentation. So there's that gap in knowledge. So for us to be able to collect it again, so we'll be able to fill that gap. You know, that is, we'll be able to do the DNA, and then you have a more accurate way of identifying them. But the observation is that, you see, there's no knowledge or scientific knowledge with the people, with the locals. So there are farming activities, so there's degradation of the habitat and all those things. So these terra typica are being challenged. There may be likelihood that in the next couple of years, if you go back, you may not even find them, because now where you find them is highly limited. Now, another major finding is, so this fish species, Fundulupacha sporo, so in the run-up to the project, I didn't have this information. But when I collected this fish and I needed to identify it, I needed to know about it. So it was then I discovered that it was originally collected in 1976 by an aquarium enthusiast, not a scientist. So he just collected it. There is even no information about where he so he didn't collect it in a scientific way. So he just wanted to sell. So, and he sold it to some European guys. So, but some scientists went to the aquarium shops and saw this, and then noticed that this, at the time, was not known to science. So they pick it up, they describe it, but there's no information about where it was originally collected. So since 1976, there has been the search for where it exists in nature. And that's the significance of this finding. So for the first time, we're able to find where it exists in nature, and we've been able to publish it in a peer-reviewed journal. So we've provided the locality, and then some other work can go on it. So these are the tilapia, so to say, tilapia fishes. So we have these four. We've run, because I work with the team, so some in Europe, some, other people are in Europe. So we have the first one and the last one, they are under what we call Coptodon species. So we've run preliminary DNA analysis, and it has established that they are distinct and they are not known to science. The second from the left, it's a Benitochromis species. We've also run an analysis on that, and its cluster, which is showing it's more related to a fish species called Benitochromis conjunctus. So the challenge with it is, the second from the right. So the challenge with it is I didn't have good quality photographs. So because the diagnostic feature, so I did more explanation. The diagnostic feature, so there's limited diagnostic feature to be able to see, although it clusters with conjunctors, but I need to establish whether it's conjunctors. If I have to establish that it's conjunctors or it's different from conjunctors, I need diagnostic feature. It was very difficult to get any diagnostic feature, so we were resting on using the photography. But the quality of the photographs I was able to get was not good enough. So going forward, I have to go back to the field, resample it, and more carefully take a good photography. So, but the other one is the Pelvicochromis is fully established. So out of the four, three are convincingly established as totally new to science. The third one, so work is still ongoing on them. So these are the explanations. So I mentioned the Procatopus. So these are the Procatopus. Work is still ongoing. So I'm working with an expert in Europe. So what we have found out about it is the group is generally having a lot of issues in terms of the identity. So there's what we call synonyms. So a lot of the original species collected, the way I will explain it, let's say it is named A, another person collects another one, and then let's say another one, and then later they will say, okay, these other ones we have collected is the same thing as the original one. So there's a lot of that synonymizations. So even though we want to identify this, then we'll find out that we need to understand the group first before we can fit our own collections. So by the moment, at least two does not fit into any of the known species. So, but again, this work is ongoing. So this is the Epiplatis. This one is entirely new to science. So these are the ones I said were originally picked from that geographical region. We have picked them again. We have collected them again. And then, so we will be the first to provide the DNA documentation for them. So these are other species collected from their terra typica. So the Ukpan and the Rubiano. So we provide the DNA. So ultimately, so maybe I will say this, because of the outstanding significance of the Sporumbeji, because at the time I was at the Obafemi Awolowo University, so I decided that I would deposit it, all the specimen, at the university. But the remaining collection were deposited at the ZSM in Germany because I work with that team. So you can't find Sporumbeji specimen elsewhere now except at the Obafemi Awolowo University and maybe in my private collection. And the remaining, and basically it's just to give credibility to the collection. So from the study, like you see, at least 7 new species were found. So which shows that there is significant level of lack of knowledge of fishes of that area. So I told you 2020, I was there just for 2 or 3 months, 2022, 2 or 3 months, and all those things. So, and if you look at the expanse of the geography, I wouldn't say I have fully was able, and it was just myself and an assistant. So it means with more time and extensive study, there will be a lot more discoveries that will be made. And then the other one is a lot of new species that were collected, and we are still working on them, running analysis. And then the gaps, so both of these reveals a lot of gap in knowledge of the area. So the ultimate challenge is that area, even if you want to conserve it or you want to utilize anything from the area, number one, you don't even have the information of the fishes of the area. And there is the challenge of anthropogenic activity even before you have information about the fishes of the area. So we are yet to understand the biodiversity of the area, but we are already losing it. So whatever advantage that we can make out of the area, it's generally now at a great disadvantage. So for scientists, so the thing is like, we have collected these species, so we need to work fast and provide the identity of the fishes that are known. And there's implication also for conservation, at least like the ones collected from the terra typica, and they are already being threatened. So we need a conservation approach to be able to salvage them. But ultimately, we need a lot more funding to be able to do more studies and take the necessary step for that area.